Welcome to this module. Before we begin, let's look at what you will learn. By the end of this session, you will understand how clinical trials progress from early laboratory research into multiple phases of human testing. You'll gain insight into how safety and effectiveness are evaluated and how these data support regulatory approval. You'll also understand how participants and whether participants, healthy volunteers, or study personnel play a key role in generating reliable results. These concepts form the foundation of modern clinical research and will prepare you for future modules, as well as real-world roles in the clinical trials environment. The purpose of clinical trials is to answer one core question. Does a new treatment work, and is it safe? Every drug, device, vaccine, or intervention must go through systematic testing before becoming widely available. Clinical trials help ensure that the public receives treatments that have been reviewed with scientific rigor rather than the assumptions of untested theories. They protect patients by requiring structured oversight, ethical review, and monitoring every stage. Without clinical trials, new therapies could be introduced without understanding who they work for and/or how well they work and what risks they take. So when we look at structure and study size, clinical trials move forward in very planned stages called phases. And each of these phases has a distinct purpose and design. Early phases involve small numbers of participants and focus mainly on safety and dosage, while later phases expand to larger populations and begin comparing the new treatment to standard of care, or existing options, or maybe a placebo. The structure of each of the phases includes their study goals, population size, study duration, data collection procedures, and their safety monitoring processes. That information is gained in one phase actually determines whether a trial can progress to the next phase. The role of participants, whether someone is a healthy volunteer or a patient with the condition that's being studied, participants are central to the entire clinical research system. Understanding the phase of a study helps participants know what types of procedures they can expect, what visits they expect. It'll help them understand what kind of risks and benefits might be involved, and see their participation really contributes to the scientific progress. Participants are partners in the research, not test subjects. Their contribution is critical to the development of new medical knowledge and improved healthcare. A well-informed participant is more confident, more engaged, and better positioned to answer questions and advocate for themselves. Let's start with one of the most critical topics in clinical research, the phases of clinical trials. Each phase, from early testing to long-term follow-up, plays a vital role in shaping the development and approval of new treatments. Whether you're hoping to work in the field or simply want to understand the process better, this module gives you the structure you need to build your knowledge. We begin with early safety assessments. These are the Phase 1 trials. In these studies, new drugs or therapies are tested on a small group of participants to evaluate safety, identify side effects, and determine the appropriate dosage. Next, we move to efficacy and approval process, phases 2 and 3. These mid-stage trials assess how effective the treatment is, and collect the data needed to apply for regulatory approval. These phases involve larger groups of participants and often include comparisons to standard treatments. Finally, we look at post-marketing surveillance, also known as Phase 4. Once a treatment is approved and available to the public, the work doesn't stop. Researchers continue to monitor for long-term safety, rare side effects, and effectiveness in real-world settings. So whether you're interested in working at a trial site, supporting data analysis, or even participating in a study, understanding the trial phases gives you the foundation to move forward. Let's dive in and explore each phase in more detail. All clinical trials begin before human subjects are ever involved. In the preclinical phase, scientists study the drug in the lab and often in animals to understand how it works and whether it's likely to be safe. If the results are promising, they move to Phase 1, where small groups of healthy volunteers test for safety and how the body processes the drug. Phase 2 expands the trial to people who actually have the condition, helping determine the best dose and continuing to monitor for side effects. In Phase 3, trials become large scale and include hundreds or even thousands of people. This phase compares the new drug to standard treatments and also provides the data needed for regulatory approval. Once approved, the drug enters a Phase 4 stage, also called post-marketing surveillance. Scientists continue to track how the drug performs in the general population. Unexpected side effects may emerge in larger or more diverse groups, and the drug's benefits and risks are continuously evaluated. Importantly, a trial can be paused or stopped at any point if it's clear evidence or harm or even success before all the data are collected. Before a drug or treatment ever reaches a human participant, it goes through preclinical testing. This phase is designed to answer a fundamental question. Is it safe enough to try it out on people? Preclinical studies use human cell cultures or animal models, such as mice, to determine how a new compound behaves in a living system. Scientists may also use test tubes or dishes to observe basic interactions. For example, they might apply a new medication to a sample of human liver cells in a lab to evaluate whether it causes toxicity. Or they may test it in mice to track how long the drug moves through the body, what we call pharmacokinetics. These tests help determine the potential toxicity, effectiveness, and mechanism of action of a treatment. Only after the treatment shows promise and does not appear to cause harm can researchers apply for regulatory approval to move into human trials. It's important to note that most drugs never make it past this phase. The goal of preclinical testing is to protect human participants by identifying risks early and ruling out unsafe compounds. Initial human testing, as in Phase 1, is the very first time a new drug or intervention is tested in human beings. It typically involves 20 to 100 healthy volunteers, or in some cases, patients with the targeted condition. These trials help researchers understand how the drug behaves in the human body, how it's absorbed, how it's distributed, metabolized, and excreted. This is often referred to as pharmacokinetics or pharmacodynamics. When we look at safety and dosage assessments, the primary goal of Phase 1 is to evaluate safety, and researchers begin with very low doses and gradually increase them while closely monitoring participants for side effects or adverse reactions. This helps establish a safe dosage range and helps to determine the maximum tolerated dose. Though these studies are not designed to test whether the drug works to treat the condition, researchers may collect early signals of effectiveness in this stage. When we look at structured study design, even though Phase 1 trials have a small number of participants, they follow a highly structured protocol. That means strict eligibility criteria, standardized data collection, precise dosing, and ongoing safety monitoring. Just like the later trials, every detail is regulated to ensure participant safety and scientific validity. The results from this phase are critical as they determine whether a drug can move forward to include larger trials. Why do you think it's important to start with healthy volunteers or participants in such a small number before testing in larger populations? After confirming safety in Phase 1, the treatment moves into Phase 2 trials, which typically involve 100 to 300 patients who have the actual medical condition the treatment is meant to address. Now, unlike Phase 1, which may involve healthy volunteers, Phase 2 tests how the drug works in real patients. This is where we start to see whether the treatment could have therapeutic value in the real world. The primary focus of Phase 2 is to evaluate how effective the treatment is. Researchers look at whether the drug actually improves symptoms or alters the course of the disease. This is done using clearly defined outcome measures such as blood pressure reduction, tumor shrinkage, or symptom relief, depending upon the study. If the treatment doesn't show signs of working, it usually doesn't move forward to Phase 3. Safety remains a top priority in Phase 2, even though it was deemed safe in a smaller group during Phase 1. New side effects or complications might appear when tested in people with the disease. These patients may be taking other medications or have coexisting conditions, which could affect the treatment's performance. Close monitoring helps researchers identify risks early and make adjustments if needed. Why do you think it's important to test treatments in patients with actual condition rather than continuing with healthy volunteers? Phase 3 trials are where a treatment gets its most rigorous test. These studies enroll 1,000 to 3,000 or more participants across multiple sites, and often across different regions or countries. The goal is to confirm the treatment's effectiveness in a diverse, real-world population, accounting for various ages, ethnic backgrounds, and coexisting health conditions. These large-scale sample sizes increase the statistical power of the study and allow researchers to detect rare side effects or variations in treatment response. In Phase 3, the new treatment is often compared to the current standard of care, not just a placebo. This comparison helps determine whether the new intervention is better, equally effective, or less effective than what's already available. This is especially important for deciding whether a new treatment is worth the cost or the risk. Phase 3 trials are frequently randomized and blinded to reduce bias and ensure objective results. Participant safety continues to be closely monitored, even at this late stage. With so many participants involved, researchers may discover rare or long-term side effects that didn't appear in the smaller studies. Data from phase 3 trials are what companies submit to regulatory agencies like the DCGI in India or the FDA in the US or the EMA in Europe when they apply for approval to market the drug. These data must demonstrate not only efficacy, but also that the treatment benefits outweigh the risks. Comparing a new treatment to the current standard of care provides a realistic understanding of how it performs in real medical practice. Most participants who may eventually receive the treatment will not be untreated. They'll receive the best available existing therapy. So this comparison gives us meaningful, practical data. In many diseases, especially serious or life-threatening ones, it would be unethical to give participants no treatment at all if an effective option already exists. Comparing new therapies to standard of care ensures patients receive at least the best proven treatment and not put at an unnecessary risk. Researchers do need to know whether a new treatment is actually better than what is already being used. A drug may outperform a placebo, but still offer no advantage over the current standard. A comparative trial helps determine whether adopting the new treatment would truly improve patient outcomes. This approach supports patient welfare, ensures trials meet ethical codes and regulatory expectations, and also strengthens trust in clinical research. Patients can feel confident that their participation is respected and that their care will not be compromised. Phase 4 trials occur after a drug has been approved by regulatory agencies and is available to the public. Just because a treatment is approved doesn't mean the monitoring stops. In fact, this phase helps assess how the drug performs in real-world settings, outside of the controlled environment of clinical trials. Researchers look at how the drug works across a broad range of patients and conditions than were tested in the earlier phases. This is especially important for evaluating long-term effectiveness, real-world dosing patterns, and interactions with other medications. Even large phase 3 trials may not uncover rare or delayed side effects, especially if they only occur in a small percentage of the population or after months or years of use. Phase 4 trials and ongoing surveillance help uncover these issues. For example, some drugs have been pulled from the market after post-marketing data revealed unexpected safety concerns. This phase helps balance the risk-benefit profile of the drug and can lead to updates in dosing, new warnings, or even withdrawal of the drug from the market if necessary. In phase 4, safety monitoring continues through adverse event reporting systems, which vary by country. In the US, for example, healthcare providers and patients can report side effects through the FDA's MedWatch program. In India, the Pharmacovigilance Program of India, the PVPI, serves this function. These systems are critical because they allow real-world users, both professionals and the public, to participate in drug safety surveillance. Reports are reviewed by national authorities who can issue safety communications or investigate further. Learning about the phases of clinical trials is crucial for students, as it provides insight into drug development and safety assessment. It helps them understand how trials progress from small-scale safety studies to large-scale efficacy testing. Understanding clinical trial phases is essential for anyone entering medical or research fields because it shows how new treatments move from early development through safety testing and ultimately into real-world medical use. Clinical trials are designed to progress in a structured, step-by-step process from laboratory and preclinical studies to phase 4 studies after they are approved. Each phase adds a new layer of evidence and helps us answer questions about safety, effectiveness, side effects, and long-term results. By learning how studies move through small safety tests to large-scale evaluations involving thousands of people, students gain a deeper understanding and appreciation for the rigor that supports modern medical advancements. No treatment is supposed to reach patients until data show that it works and it is safe. Understanding clinical phases also helps students evaluate research critically. Once you know what type of information each phase should produce, you can judge whether a study is well designed and whether its conclusions are valid. The knowledge prepares research clinicians and administrators to contribute confidently to the medical field, whether they are collecting data, coordinating studies, reviewing safety reports, or working directly with patients. Knowing the clinical trial process is not just about passing a course. It's a foundation for making informed decisions in science, medicine, and patient care. Well, thanks for joining us for this module. I hope you're feeling excited about the possibilities around getting involved in clinical research. Don't worry if this is all new to you. You'll gain confidence as we move forward together. I look forward to seeing you in the next module. In our next module, we'll explore the different roles in a clinical trial team. From investigators to data specialists, you'll learn who does what and how these roles work together to bring treatments to life.